In this final video of chapter 21, we're going to talk about biochemistry. We're first going to take a look at the structure of an amino acid, which is the smallest building block of proteins. This is the general Lewis structure for an amino acid here. And importantly, we get the name amino acid because it contains an amine functional group and it contains a carboxylic acid functional group. So that's where we get amino acid from. And there are going to be differing R groups that are attached to this carbon in the center. And as you change the R group, that's going to change the identity of the amino acid. Now, there are 20 common amino acids that we encounter in our everyday lives. And some of these amino acids are going to contain nonpolar R groups. And here are a few examples. So the R group could be something as simple as just a hydrogen atom in the case of glycine, or you could start talking about much more complicated R groups, right? When you have like benzene rings and stuff like that. I am not going to make you memorize the structures of amino acids. I'm just putting up these structures here to allow you to see different amino acids result from having different R groups that are attached to that backbone there. Now, 11 of the amino acids have polar R groups, right? And those are characterized by, when you look at, let's say, tyrosine, you see an OH group here, right? And so that will have hydrogen bonding IMFs. You could even have sulfur present in your R group, or you might have carboxylic acid groups like this. And so that will give you ion dipole interactions in aqueous solution if you were talking about, let's say, glutamate. Again, I'm not going to make you memorize all of these structures. I'm just putting this information up here just so that you can see the variety of different amino acid structures that you can have. Now, one thing that you will be expected to know about is how you can begin to make proteins by performing condensation polymerization between two amino acids. So here's one amino acid, and here's a second amino acid molecule. And the carboxylic acid functional group on one of them can react with the amino acid functional group on the other. A water molecule gets eliminated, allowing these two amino acids to be linked together by an amide or a peptide bond, just like that. Then you can begin repeating this reaction, extending the molecule going to the right by reacting the carboxylic acid group here with another amino acid group. You can extend it going to the left by reacting this amino acid group with a carboxylic acid on a neighboring amino acid. And that allows you to begin generating a protein, a big polymer made up of amino acid building blocks. Let's talk a little bit about protein structures. So we're going to talk about three types of protein structures here. The first is called the primary protein structure, which tells you the actual sequence of amino acids that are used to make up the protein. There are a couple different ways that you could represent a primary protein structure. So one way that you could go about doing it is by drawing the full Lewis structure that you would have for each of the amino acids and have them all linked together with their peptide bonds. Another way that you could represent the primary protein structure is by listing out all of the amino acids that were used to make the polypeptide here. So in this case, we have an alanine, a glycine, and a phenylalanine, and so we'll just say alaglyphe, just like that. Importantly, when you talk about what the sequence of your amino acids are, you have to always name the amino acids and list them going from the nitrogen terminus to the carbon terminus. So what I mean by that is you always have to draw the molecule in a way where you put the amino acid side of the molecule on the left and put the carboxylic acid side of the molecule on the right-hand side. And then you would list the amino acids going in that order, right? So that's how we would go about naming the amino acids that are in this primary protein structure, always going from N terminus on the left to the C terminus on the right. Now, if there are only 3 amino acids in your polypeptide, it might be convenient for you to draw the full Lewis structure showing all of the atoms. But the problem is that proteins are made up of much more than just 3 amino acids. There are a lot of amino acids that make up a protein. And so it can start to get very cumbersome and take up a lot of space to draw all of these atoms. And so there are quicker ways of representing a primary protein structure. Another way is by just using the alphabet of amino acids that we have. So each amino acid is designated with a letter, and then you could just use those letters to indicate what amino acids were used to make the polypeptide here. Again, this goes from here's the nitrogen terminus all the way through to your carbon terminus. And then that's a way that you could represent a sequence of 28 amino acids without having to draw every single atom here. Every letter here represents a different amino acid. I'll also say here that I'm not going to make you memorize the alphabet of amino acids, so you don't have to know what D or V or E means. You just have to know that a primary protein structure tells you the actual sequence of amino acids, and you also have to know that the primary protein structure is always going to be listed going from the nitrogen terminus to the carbon terminus. That's what you need to know from this slide. Next, we'll talk about the secondary protein structure. Secondary protein structures give you information about the shape of the chain of amino acids at relatively short range. And the structure that the protein will adopt will be determined based on intermolecular forces. Generally, you're trying to maximize the extent of hydrogen bonding, and so to do that, you'll adopt one of two types of secondary structures. The first is called an alpha helix. And so in this secondary structure, all the atoms are actually being drawn in order for you to be able to see the hydrogen bonding interactions that are occurring. So by this coiling up into a helical shape like this, that allows neighboring amino acids to form hydrogen bonding interactions with one another, right? So you have this hydrogen atom from this amino acid that is making a hydrogen bonding interaction with this oxygen atom from another amino acid that you have, right? And by them forming into this helical shape, it's twisting around among itself, and so that makes it so that it's possible for these to see each other and maximize their extent of hydrogen bonding. So this is one type of secondary structure called an alpha helix. The second type of structure would be a beta or pleated sheet. So this would be when you have hydrogen bonding between two neighboring chains. And so these two chains kind of form this sort of zigzag pattern that forms, again, called a pleated sheet. And then the two neighboring chains try to maximize their IMFs with one another. So here again, you can see here's a hydrogen atom and here's an oxygen atom, and they're doing a hydrogen bonding interaction just like that. Oftentimes, when you start talking about relatively large proteins, it could become kind of cumbersome to draw all of the atoms in here. And so sometimes you'll just see it represented by a ribbon structure like this if you're talking about an alpha helix, or like this if you're talking about a beta or pleated sheet. You don't have to draw all of the atoms to represent the structure that you're trying to talk about here. The important takeaway thing from this slide and what you need to know about secondary structures is that they tell you the shape of the protein chain over a short range. You have to know that there are two types of secondary protein structures, one called an alpha helix, where you have hydrogen bonding within the same chain, and beta or pleated sheets, where you have hydrogen bonding between two or more chains. And lastly, you need to know that the type of structure that it adopts is determined based on what helps you to maximize hydrogen bonding interactions. The third and final type of protein structure is what's called the tertiary structure, and this tells you the overall structure of the protein at long range. And so we're just zooming even further out when we start looking at the tertiary structure here. And so in this case, since we're looking at the whole protein, you're not going to show all of the atoms here, right? So this is just showing the ribbons. So this is basically just showing an alpha helix, right, secondary structure, and then you zoom out to see the tertiary structure to see how the alpha helix folds into the overall tertiary structure of the overall protein here. Now, this structure is going to be dictated by what maximizes IMFs. It's going to be dictated by covalent bonding interactions. And so that will all play a role in determining the tertiary structure. The key takeaway message from this is that, and what you need to know from this, is that the tertiary structure tells you the overall structure of the protein. And there's a lot more that you can get into surrounding protein structures and biochemistry. There are a lot of other ways that you can see organic chemistry applied in the area of biochemistry when you start talking about things like sugars and carbohydrates, and when you talk about lipids. And it's a really fascinating area, but that is an entirely different class from general chemistry. And I recognize that many of you are concurrently taking Intro Bio this quarter, and so you've already had a unit where you've learned about all of that. And so I'm going to spare you the redundancy of hearing all that information presented again. And you probably heard it from someone who is a lot better at explaining it and knows it a lot better than someone with a PhD in physical chemistry. So this is going to be where I end the discussion of biochemistry. And this is where I'm going to end the discussion of Chapter 21 in organic chemistry. So at the end of this presentation, you'll see the bulleted list of topics that we discussed in this chapter. You can see this and see this and use it as a study guide if you want. Right? We really did talk about a lot here in Chapter 21. And so definitely do recommend taking some time to go back and revisit a lot of the naming examples that we went over, all the stuff that we talked about with functional groups, everything that we dealt with in terms of polymers. There were a lot of things that we discussed here in this chapter. Now up next, we're going to start Chapter 20 when we talk about nuclear chemistry.